Hi, everyone. Cool. Okay, great. Thanks, everyone, for joining on the webinar. My name is Carol. I'm a field application scientist for Vizgen, and I'm just going to give you guys a few words of introduction on behalf of Vizgen before leaving the floor to our wonderful speakers today. We are a spatial transcriptomics company, and the platform that we support is called MERSCOPE. So just to put this into some context for people who are not familiar with the technology, it's a platform designed to make MERFISH available to users. MERFISH stands for multiplexed error robust fluorescence in situ hybridization. So really briefly, it's a combination of classical single molecule FISH for direct RNA quantification in situ, combined with a proprietary barcoding system to enable really high multiplexing. And this enables to keep single-cell RNA detection with high multiplex level while conserving high accuracy and consistency. So we are putting all of this into an easy-to-use, fully automated platform with support from panel design to use to data analysis. So just really quickly, a few features of the instrument. We currently are able to support panels of genes from hundreds to 960 genes detectable in a single sample. The resolution that we get on the image at the pixel level at the end is 100 nanometers, so really enabling you to go get some insight at the subcellular level. And we conserved a really high sensitivity due to direct single molecule hybridization inherited from SMFISH. But something that I think will be really highlighted during the presentation today is the flexibility of the platform, because we currently support fully custom panel design on any species that has an annotated genome. So we've got plants, we've got flies, we've got mice, non-human primate, xenopus, zebrafish, anything is possible. And currently, with our newly released platform, the MERSCOPE Ultra, we're able to image up to 3 square centimeters of tissue per run. So that's over several hundred thousands of cells per sample. So just a few key concepts to understand the MERFISH chemistry before we delve into applications. The key pillar of MERFISH is combinatorial labeling. So that means that before we even get started with the hands-on experiment, each of the genes in your panel is given a binary barcode with zeros and ones that each contribute to the presence or the absence of a fluorescent signal on the sample as detected by the microscope. So as the run goes on, the microscope performs sequential imaging. So with imaging cycles, we record raw images that are stored and then decoding to compare a final barcode to the expected barcode of the gene. And the last key strength of MERFISH is, as the name indicates, the error-robust barcoding enabled by the bioinformatics behind it. And by the way, the barcodes are structured together. So this is really what brings us to reliable high multiplexing. If we just look at the molecular aspects of it so that you can understand the presentations afterwards, we hybridize usually 25 to 50 MERFISH probes to each of your RNA target. And as you can see, they have little colored here overhang areas. And these overhang areas, once they are acquired in the instruments, they hybridize to fluorescent readout probes. So this is what generates the fluorescence, and this is what's cleaved and then rehybridized over the cycles to build the sequential imaging. Here, just a few points for you to understand. The workflow itself starts with hybridization of the probes, which can take 2 days usually. Then we have a key step of clearing, during which we embed the gel into the tissue into some gel, and we digest all the proteins and lipids away. And then the actual imaging with the fluorescent reporters is done in the microscope. Today, we are really happy to welcome 3 co-workers from the Earlham Institute, which is based in Norwich, Norfolk in the UK. It's a research institute focusing on innovation and training, mainly around genomics. And we're first going to have a talk by Ian Macaulay, who is the group leader, the technical development group leader and he's going to tell us a little bit about his experience setting up spatial transcriptomics in Vizgen at the Earlham Institute. And then we're going to have Ashley Lister, who's a senior research assistant in his group, who's going to explain how she optimized plant tissue studies with MERSCOPE, both from technical considerations to analysis with some beautiful data. And finally, we'll have Sonia, who's a postdoc in the Wojtowicz group. And she will tell us a bit more about how she works on understanding challenging cell types with MERSCOPE, and she focuses on megakaryocytes. So really happy to leave the floor to these really interesting speakers, and I'll see you guys later for the questions.
Are my slides showing okay?
Yeah.
Excellent. Okay, cool. So thank you for the introduction, Carol, and thanks for having us here today. So I'm just going to talk a little bit about our background working with spatial transcriptomics and our experience to date with the Vizgen platform before handing over to Ash and Sonia to talk more about the biology and the kind of observations we're making with the platform. So as you mentioned, I'm a technical development group leader here at the Earlham Institute. And the Earlham Institute is a UKRI funded research institute in the east of England in Norwich. And so our remit is really data-driven biosciences and genomics across the life sciences. So we work on anything from bacteria to plants to human and mouse. Any species goes really. So we have to have genomic tools that enable us to address questions in a really wide area of activity. We also run, as part of that, something called a National Biosciences Research Infrastructure in transformative genomics. And this also, in addition to our research activities, enables us to provide support and service and training to the wider UK and further afield biosciences community. So when we get new technologies, we're quite good at sharing them with other people and letting other people sort of get some experience with them too. So, as I said, we work on all sorts of things from biofilms right through to human brains. And so for about a decade now, we've been working with single cell technologies, and that's been very exciting in terms of enabling us to generate things like this, which I'll just put my pointer on. Things like this. So you know, UMAP plots showing us the complexity of the different cells that make up our biological system. But as satisfying as that's been, there is a lot of dissatisfaction with that, because we don't really get a picture of how the organism functions, how when we put all of these individual cells together, we get things like tissues and whole organisms emerging and functioning. And so that's a key thing we've been feeling like we've been missing out on for a long time in single cell. And so in many ways, we've been sort of pursuing a spatial transcriptomics journey before the advent of the sort of current technologies, mostly relying on low input RNA sequencing, initially looking at chunks of tissue, literally sectioned with a razor blade into sort of millimeter squared areas and generating RNA-seq libraries from those, and then gradually reducing that down to look in the wheat spikelet, which Ash will talk about a little bit later, at sort of different regions of small tissues, but still not single cell, just kind of sort of 0.2 to 1 millimeter squared pieces of tissues. And then down to in the chick embryo, we've done a really nice study where we took serial sections through a developing chick embryo. And from each of those 20 micron sections, about 180 of them in a row made low input single cell RNA sequence libraries using kind of an RNA tomography approach. And so all of these approaches have kind of highlighted to us in different systems that the spatial organization of gene expression is something we can observe and we want to observe, and that has really potential to generate novel insights about how tissues work. But again, none of this is at single cell resolution. And then in 2021, for 2020, obviously, spatially resolved transcriptomics was announced as method of the year. But what does that really mean? There's so many different approaches. And I'd really advise, if people have time and are interested, obviously look at the method of the year stuff from 2021. But there's a lovely paper, an associated GitHub, that is this Museum of Spatial Transcriptomics from Lior Pachter, that has a kind of an archive of all of the different developments in spatial transcriptomics, but also an archive of all of the different studies that are being published and the kind of platforms they use and the technologies and techniques and the systems they're looking at. And that's a really good place to get a sort of an overview of what people are doing, and so an overview of what technologies are out there. So we were kind of looking at these things and trying to make our own mind up about which platform we were keen to pursue. So our key considerations in choosing a platform, as we've mentioned, we were very keen to work with stuff that was species independent. We couldn't really work with something that was mouse and human only. So that was our kind of our first consideration. The resolution, we'd obviously explored other options where perhaps the resolution wasn't single cell. I think it was key to us, having worked with single cell genomics for so long, that we would still maintain that single cell resolution. We knew again from the work that I mentioned previously, the areas of interest we'd like to be able to explore are not millimeters squared, they're sort of centimeters squared. We want to look at reasonably large pieces of tissue. And again, the sort of number of genes we'd like to look at in experiment, ideally we could look at whole transcriptome, but in reality, most of the things we're interested in, we could refine down to panels of a few hundred genes, maybe more. And of course, the constant concern with all of these things is the instrument and consumables cost. So those are the things we had in mind when we were starting to explore a new platform. And so looking around, we sort of had a look at the different platforms available, but we looked at the Vizgen platform through that lens and saw that we could see, we could do fully customized panels. There was no species restrictions. The operation, or the panel size operated between 140 and 1,000 genes per panel, 1 centimeter squared analysis area, and it had our single cell resolution, even arguably subcellular resolution, and comparable pricing, and of course no sequencing costs. This is the sort of thing to keep in mind from a sequencing perspective. We're always used to running our cells on a device or generating single cell RNA sequencing libraries, and then having to pay additionally for sequencing. Whereas obviously with these in situ approaches, there's no sequencing associated. So at least all of the cost is contained with what you do on the instrument. So we received our instrument in April 2023 and ran some mouse brain pilot data with one of the FASs. And this was really the sort of point at which we realized we'd made a good decision. This is a mouse brain, which I think none of us directly work on mouse brain, but we're kind of blown away by the quality of the data, the scale that we could make these observations on. So you've got a whole section through a mouse brain, and we could identify different cell types in that. And what was really satisfying was then to start looking at the different clusters present in that data, and see that we could visualize those clusters spatially, which is great, but also look at marker genes that are associated with those clusters and see which marker genes are expressed in different places. I think the highlight of this little piece of work was going through the Allen Brain Atlas and trying to find the same gene in a section that's been done by a completely different method, and just sort of validating what we observe on the Vizgen with someone else's data. And showing that the same gene is expressed in the same area and the same approximate section of tissue. And sort of that just gave us real confidence that the kind of things we're seeing with the platform are real, and that actually we can make really quite detailed biological measurements with this. What's amazing is when you generate these plots and start zooming in very closely on them, you really get a sense of the scale of the data you're generating. So as I said, we sort of offer a service, or at least a collaborative service to the community, and we've had lots and lots of inquiries about what we can do with the platform, and some of those are progressing to full-blown projects at this stage. Everything from plant to human, mouse, fish, hamster, chicken, mouse, lung and bone marrow, and I think Ashley will talk mostly about our work in the plant area, and Sonia is going to talk about the work that she's been doing in and around hematopoiesis and mouse lung and potentially bone marrow. But really what we're finding is that the interest that the platform is bringing in is from a really diverse audience, and that's really good for us. I think it works extremely well for us. And it's quite satisfying to see lots of non-model organisms as well as model systems being presented as things we would pursue with this platform. The other thing it's done for us is given us really nice new opportunities to collaborate and engage with the community. So we ran, I think, probably the first spatial transcriptomics hands-on workshop in plants earlier this year. So Ashley organized this with a lot of support from Carol. And it was, again, really, really good to see the sort of rapid adoption in the plant community of spatial technologies. And I think it fits really well with a lot of questions in plant biology. But to have a heavily subscribed and active meeting so early on in embracing a new technology was really good. I think really exciting for us. So the considerations that we've had following our platform implementation, the lessons we've learned along the way, I think that there are definitely challenges with getting started with this. I think the main one we've encountered is obviously that sectioning and sample prep are key. We found working with tissue-specific experts is the best way to address tissue-specific problems. Overall, for something that's coming into a sequencing laboratory where we might not necessarily have been handling sectioning all day, every day, this is kind of a new experience for us, and that's been a learning curve. Panel design, it takes a surprising amount of time sometimes to come up with the gene panels. It's easy to maybe come up with your 70 or 80 favorite genes, but then making that to 300 sometimes is a little bit hard, because you will add in lots and lots of extra things and get a panel of 500 or 1,000 quite easily, and then have to bring that back down depending on your budget. The other major observation really has been around the data, the amount of data the platform generated. It generates a lot of data, which is fine. It's comparable to sequencing, but data management is a key part of your sort of activities and understanding what you're going to do with the instrument, and what the kind of the outputs are, and where you're going to put that data. As we run it as a partial service now, it's scheduling is an issue. So we do have to think about how many runs per week we can manage. On average, we see about one run being feasible per week with our current staffing and instrument sort of setup. But that just means managing people's expectations about turnaround is fairly important. In terms of costs, these are expensive experiments, but they're the same sort of price as a single cell experiment. The budget outlay is significant, but that just means it's important to get it right. And I think we spend a lot of time making sure that the preliminary work has been done around projects, and that people are interested in running the verification kits and other things before they get started with a really huge project. And in terms of informatics, we've had to learn lots of new things. Everything from cell segmentation through to sort of how we visualize the data is just a little bit different from standard single cell work. So there's lots to learn and lots of new things that we're encountering in that area. So those are sort of considerations, things that came to mind that we've encountered during our establishing the platform. But overall, I mean, there's been lots of work to get this up and running, but I think, and I hope you'll see from the subsequent presentations, that it's been absolutely worth it. I think the kind of data we generate is very captivating. It's very easy to engage with sort of non-expert audiences with, as well as demonstrating really interesting biological phenomena. I think it's a fantastic addition to our sort of capability, and I'm really enjoying seeing the data that comes from it. So I'll just finish up by saying thank you. If anyone would like to get in touch and work with us on our platforms, Ashley, Vanda, and Andy are sort of our platforms team, our platforms experts on this. If you'd like to send us an email, please do get in touch, and I'll leave it there. Thank you. Shall I stop?
I'm going to share my screen now. Ian, can you just let me know if you can see that?
Yeah.
Thank you. Okay, so hi everybody. My name's Ashley Lister. I'm a senior research assistant in Ian's group. Yeah, we mainly specialize in methods for single cell and spatial technology now. And I'm going to talk to you today about how we're applying the Vizgen MERSCOPE method to some plant tissues, which I guess is kind of unusual in the spatial world. Just to give you a little bit of background, I'm not a microscopist. I do nothing with images ordinarily. I certainly don't do any histology. My background is anything that's sort of directed towards a sequencer. As I say, we focus on single cell methodologies, and so that's often cell isolation for a fax into plate-based methods, and from then on a lot of automation. Otherwise, droplet-based techniques, and I focus on these techniques for the applications in the plant world. So it's been a big learning curve, having to learn a lot of these new skills ready for spatial transcriptomics. So ordinarily, when we conduct single cell isolation for sequencing in the plant world, The main problem that we have with plant cells is that they have this thick cell wall to contend with, which means that the lysis properties are particularly difficult. So the first stage is to digest away the cell wall. And at this point, we are left with what's called a protoplast, a spherical cell that is left with a thinner membrane, which acts actually a lot like a mammalian cell. So is then it can be inputted into single cell methods that have actually been developed really for mammalian systems. We can go further than that and digest away the rest of the remaining material and be left with nuclei. So either are appropriate for RNA sequencing, but there are advantages and disadvantages for that, as you will see. Plant cells tend to be a little bit bigger in some cases not, of course. Their shapes can be very unusual, and I know that that's not necessarily a plant thing, that can be in mammalian systems too. But the main trouble with plant cells is that they have these awful cell walls that are really, really tricky to work with, and that makes them particularly difficult to dissociate. Even at protoplast stage, they can be quite sticky. So there's definitely challenges to doing single cell sequencing. So why do we require spatial transcriptomics? Well, it has the advantage that we don't need to do that dissociation. We can keep those cells in situ. Of course, there's then challenges to doing the sectioning, but it means that we can keep those cells in situ with the advantage of maintaining the cell's microenvironment, maintaining the neighboring cells surrounding that environment. So who might be talking to who, different sort of molecular pathways can be dissected. It allows for different cell types, different shapes, and larger cell types. So that's a real advantage. It means that we have no limitation in that at all. We're particularly liking that a lot of the spatial methods, including Vizgen, allow for fixed tissue samples. So that's particularly good for time course experiments. And with the Vizgen MERSCOPE, it allows us to target the genes that we're interested in, and ignoring a lot of the background RNA sequencing reads. To talk to you about how I go about processing plant tissues for Vizgen MERSCOPE runs, I just need to explain a little bit about the workflow, and Carol's just introduced that very briefly. So very roughly, the biggest stage that we found to help you optimize your runs for MERFISH is that you need to spend a lot of time learning how to fix, embed, and section your sample type. It's no good picking up a protocol and expecting it to work in your sample type, even if it's a similar sample type. Different developmental stages might perform a little bit differently. So spend a lot of time with that and check the quality. So once you have some tissue onto slides and you're happy with the RNA and the integrity of the sample, we go forward to a process called verification. Verification kits can also be custom made. And so with the slides that I'll show you next, the verification kit has been customized for plant tissue. So we run a housekeeping gene and just check the RNA looks good and the tissue morphology before we spend a lot more money and commit to full probe runs. At this point, I'd really like to introduce Katie Long. Katie is my collaborator over at the John Innes Centre. So actually they're next door to us in the building on the Norwich Research Park. And Katie is a PhD student who's really, really talented, particularly in the sample preparation. So getting the tissue onto the slides. So for this, there'll be a phase of plant growth to the point where the tissue is at the correct developmental stage for harvesting and dissecting out. Her tissue has been fixed in PFA before it's embedded, ready for sectioning on the cryostat, and some images here in the bottom. And you can see in these images just how many tissue pieces that are in one block, which just shows how talented Katie is at doing this job. Just a few little tips and tricks that might help you for sectioning plant tissues. The cryostat temperature might be a little bit different if you're ordinarily used to sectioning animal tissues. If you remember, cells in the plant world actually have a lot greater water content. So you need to adapt your temperature for that, so that you're not bursting your cells just from freezing. But it might help that effect if you get your dehydration step right. And the samples I'm going to talk to you about today went through a sucrose dehydration phase. The samples are fixed, but that might be an option for you. It might be better that they don't. So you need to play around with that a little bit. And just the main thing that we found when we're starting off our Vizgen journey is that when you're sectioning or learning to section, you need to be practicing this, and not just every so often. You need to be doing it on quite a regular basis to maintain that skill. I've been calling it an art form, actually. It's not really a skill. In the video, I have a bad slide. Don't produce slides like this. It's not going to work for MERSCOPE, but I think that's maybe a little bit more obvious. But basically, when you're doing your sectioning, it's really good to have a microscope right there next to the cryostat so that you're constantly able to check the quality of your tissue. So the things that we're looking at is that there's no pieces of tissue that are peeling away from the main bulk. Your cells look nice and plump and correct in their morphology. Again, you would need to know your tissue type to know that that is correct. Adherence, so clearly in the video, this is particularly bad. You actually have lots of pieces that are flapping around in the solution. But if you should basically have tissue that is on the same Z plane, so you should never have to refocus if you're moving around the slide there under the microscope. For plant tissues and some mammalian tissues, you're going to need to check what the autofluorescence is, so the natural fluorescent properties in your tissue type. And in plant cells, that's normally in the cell walls, where there's a lot of lignin deposition. And I found that actually, even just within a few days developmentally, there is a lot more autofluorescence that has been found. A good thing to do at this stage is to check the DAPI staining, which is an eventual stage before a MERSCOPE run. So it's a good thing to do at this stage as well. And one thing that Vizgen recommends is that you can actually do an RNA extraction on just a few of the sections from the block that you would eventually land onto a Vizgen slide, just to check that your RNA is of good quality and it has nice long fragments. Just as an Excel spreadsheet, I just wanted to give you an overview of what the workflow looks like and how long it is. So it can take us about 2 weeks, essentially, from the point where Katie is doing her sectioning to where we have data to be able to visualize. So it is fairly lengthy. And so this is what one sample would take me this sort of long week. Just to Sonia will maybe talk to you about this in a minute, but just before the probe hybridization on a Monday morning, first thing on a Monday morning, those of you that are doing animal samples will need to fit some cell boundary staining and perhaps protein staining as well ahead of this stage. So Ian alluded that we're sort of doing 1 a week. For big projects, we have managed to do 2 a week, but we definitely don't promise this. And this is how I go about batching samples for any of you that are in sort of a core facility that are going to be running these types of samples. So I'm running batches of 2, but on a Monday morning, I'm doing a run in the afternoon, and I'm setting off my next batch of 2 on that Monday morning. So that's kind of how I schedule it. And sometimes that works, sometimes it doesn't. It depends how long some of your samples are taking to clear. As I mentioned before, plants have this inherent autofluorescent property. And the way that Vizgen can overcome this is they provide a little device called a photo bleacher. Basically, it overexpresses the UV light onto the tissue in their Petri dishes on their slides, and it dulls the autofluorescence specifically in the DAPI channel. So this is what the wheat spike looks like in that central picture. There's a lot of autofluorescence in the cell wall, and on the right is a wheat grain image as well, where we're getting actually all of the colors of the rainbow in the seed coat. So that's particularly difficult. But the photo bleacher works well to dull that down. Just to fit this back into the workflow, the next stage would be probe hybridization. I've included an image of how we go about doing this. So we do an initial wash with formamide just to fix everything down, and the probes are hybridized in their Petri dish, and they are incubated for 48 hours. There's a little video here of how we go about doing gel embedding. So we drop a little bit of polyacrylamide gel, and this is all provided in a sample prep kit from Vizgen. Cover it over with a cover slip, take away the excess. And what that does is it just gives a little bit of thickness of gel to cover up the tissue to maintain its integrity as it goes through the next stage, which is called clearing. Poly A tails are captured during this phase as well. So it fixes the probe spots and the gene expression spots in their spatial locations, ready for the next stages. As I say, the next stage is clearing, and this is particularly difficult for plant tissue. Those of you doing animal tissue, this will be a pointless picture. You won't see anything at all under a microscope, actually. It will completely clear. But for plant tissue, we found that it works well on the cytoplasm for sure. It clears away the proteins that are in the background there, but the cell walls are actually retained, but it doesn't seem to make much of an effect as long as the autofluorescence is dulled enough. So I would suggest for plant tissue, certainly for the wheat spikes, you definitely need to do it for at least 2 days. And we go through the non-resistant workflow. But the best is actually for 4 days. So actually we clear it over the weekend oftentimes. I've included the next stage, which is getting ready for instrument setup, where we stain for DAPI and poly T. Just included some images of what that looks like because they're particularly pretty, I think. And then this is how we set up for an instrument run. The circular Vizgen slides are fitted into this metallic adapter called a flow chamber, which has these aqueducts on the side, so the tubings, which will be connected to a fluidic cartridge that is in the other piece of the instrument that's under my arm there in the video. Okay, so I'm getting down to the use case now. So this project was looking at wheat floral development. So this is sort of the problem slide, why we need to do it, what we're interested in. So those of you that are not familiar with wheat, wheat is a domesticated grass. It's grown all over the world, actually. And it's especially economically important because many people will be eating wheat products every single day. I'm guessing that a lot of you will have already eaten them this morning or this evening, depending where you are in the world. So the wheat spike is this piece on the very far side of the screen here. It's the inflorescence or the cluster of floral regions at the very apex of the plant. So the spike, or the ear, as it's called, produces grain, which is the agronomically interesting part of the plant. The grain can be then taken forward to make many food products or drink products. And in the base, there is a particular problem in domesticated wheat where there is a redundancy of grain set. So it often doesn't bear grain, rarely bears grain. So the group that I'm working with at JIC, so in conjunction with Katie Long and her group leader, Christopher Wowie, they're trying to look to see if there's any gene regions and gene reasonings for this redundancy in the base of the wheat spike. To put this into context of how old these plants are, they're very, very young. We're looking at what's called a double ridge stage, when the plant makes a decision to change its developmental trajectory from being a stem or a leaf, so a vegetative stage, to a floral stage, so to create a grain floret. And that's what they look like in the top there. They're very, very small. Just to take you back in history a few years ago, I worked with Katie's predecessor, Anna Backhouse, so also formerly of JIC. And they took similar time points and sample types. And this was before Spatial, clearly. And what we were trying to do is to look at differential gene expression in the different regions of the gene as we worked our way up. So we took micro dissections. They were split into 3 micro dissections of the basal, central, and apical regions. And we ran it through a protocol that Ian developed called GNT-seq, which stands for genomic and transcriptomic sequencing, to create actually just RNA sequencing libraries for the different micro dissections. So this is a single cell protocol that we run routinely in the lab. It's fully automated. but it has been set up for single cell sequencing. So it's particularly good for low input, so small cell counts. And what we found was hints that there is definitely something going on in that basal region, which is in opposing effect to the apical region. So we knew that we were onto something here. And these were the kind of just overall gene expression counts that we were finding within one spike and between those different microdissections, but actually not very many at all between the different developmental time points. So with this as a result, we knew that we needed to go to greater resolution in one spike. And this is how we've hit on the requirement for spatial transcriptomics. Before I show you some nice pictures, I just wanted to explain a little bit more about the experimental design for this, because I feel like it's particularly useful for some people in the audience. The way that this group went about probe design was that they used the low input RNA-seq results, so these little microdissections, to inform and assemble the probe set that they were going to use for the spatial transcriptomics. Often people ask me if it's a requirement to have single cell sequencing results, and I don't feel like it is, but it does make it a lot more powerful. But I'll show you why it doesn't necessarily need to be the case later on in some of these slides. The other good thing about their probe design is that they actually shared the cost with another group at the John Innes Centre. Katie and her group had 200 of the 300 gene panel, and 100 was designated for a different group for their project and their research interest. So that might be of interest to people in the audience. It's another alternative. But the good thing was that they were both studying wheat, so they could use the same custom verification gene. They can use the same housekeeping gene. A little bit more about experimental design. This group were able to group multiple technical replicates onto one slide, different time points onto one slide, and actually different mutants onto one slide. But this is fairly unique because of the size of the samples that they were able to do. So just to put you into context, we have a viewing window of 1 centimeter by 1 centimeter. We don't have the ultra, which is the 3 by 3. So yeah, that just shows you how small these samples are. So this is the result of us going through this verification kit. So just to remind you, that is one probe actually it's 2 probes, designed against one housekeeping gene. So wherever you're seeing a spot in blue or in green, you're seeing that gene being expressed in that cell at that time point. And actually it's colored turquoise because there is actually 2 probes on there. And the mixing of the 2 means that we have good co-localization of them. So it means that it's been very, very specifically hybridized. So we knew that we were able to proceed to the full probe runs at this stage. And this is the effect of applying 300 probes, so the full MERFISH run on these wheat spikes. And as you can see, it's completely littered with gene expression. We've got really, really good hybridization. The RNA must be very, very happy going through this process. So we were absolutely thrilled to get this result. To go back to workflows, I'm sorry that there's so many of these workflow slides, but the data workflow is a little bit different for plants. When you run animal tissue through the Vizgen MERSCOPE workflow, you're able to use cell boundary staining to mark out the different cells and be able to segment actually on board on the instrument. So your data comes pre-segmented. For plants, that's not the case. We only have DAPI and poly-T to work off of, which is nice in the lab. We have less to do, but for this, it takes a little bit longer. So we have to utilize a tool that Vizgen provide called VPT, so Vizgen Post-Processing Tool. And this is a Linux or Python-based algorithm that is running through something called CellPose or CellPose 2. And what that's doing is it's taking the DAPI and poly-T stained images and defining cell boundaries for us in an automated way. But it's worked fairly well for our tissue. And just to put it back into context, we're then up to speed with the rest of the mammalian systems, and we can proceed in the same way. And Vizgen have a really good tool on Google Colab, which takes you through the Scampi pipeline, which some of you, if you're in single cell worlds, you'll be fairly familiar with that type of analysis. So we can produce things like cluster-based analysis or any heat maps, any differential analysis with the different regions of interest. So this is the effect of putting CellPose on top of our DAPI and poly-T stained images. So it's working fairly well. There's definitely some regions that need to be defined. I think everybody in the spatial world knows that these pipelines need to be worked on a little bit better, but as I understand, CellPose 2 is working a lot better, and there'll be more out there. And so once we've got our cell segmentation, we're able to define more metadata. So we're literally able to count the number of spots in there, the different colors of the spots, which are representing different genes being expressed. And that's really, really informative for the downstream single cell analysis. So we're now able to go to single cell analysis and that type of resolution rather than just having pretty images. So yeah, at this stage, we're able to have sort of cell clustering. We're able to cluster the cells or the expression domains by their gene expression, so what is being expressed and how many of them are being expressed in their different domains, and we're able to overlay those cluster colors back onto the full axis view, and as you can see, we're getting really nice typing specificity across the whole spike. I haven't shown you all of the cell types across the whole time points that we've checked, as this is in publication. It's due to come out in a few weeks. I've included this slide in here, one, because it's very beautiful. You get a lot of beautiful images out of Vizgen, but also to show you just at this high resolution, just how specific our gene expression hybridization has been working. We're blown away with the results of this. It's really worked particularly well. I think this tissue type in particular is fairly good. It's quite soft tissue, so that's going to work well for you if that's something that you're interested in running on the MERSCOPE. And just how well it's worked if I overlay the cell types or the expression domains on top of that image. This is to introduce you to the MERSCOPE Visualizer tool, which is the software we can use to have a look at the images, but also to have a look at the metadata, so the different genes that we're looking for from our probe set. It'll give us things like volumes of the cells. Once we've done the segmentation, we can make heat maps, that kind of thing. We can still have our Leiden clustering map on top of there. So it's a really powerful tool, and I'm particularly impressed with it, especially comparing it to some of the open source ones that are out there. This is maybe a more unusual way of analyzing things that Katie's been doing recently, ready for the publication clearly, where she has created regions of interest, which you can do in the Visualizer tool, to create different sets of data for the different bins. So what she's done is she's created vertical lines in certain increments down what this is called a spikelet. So this is one of the bulges along the spike, to look at the expression just across one spikelet. So really, really high resolution gene expression, differential gene expression. Just in case that's of use for any of you for your analysis, that's some of the powerful things that you can do with the Visualizer tool. And just to go back to the start of where we started with those microdissections and the RNA-seq that we got from those, we're able to sort of sell overall gene expression counts. We're able to tell what genes were in there, but not what cell types they were coming from. And this is the resolution we're at now. We're able to go to single cell resolution across a whole spike, which is incredible. Really high specificity, and we're really thrilled with the results. So it's worked really nicely. And this is just giving you the overview of all of the time points that we were looking at. And you can see the little baby spike there in the end, just how messy and undeveloped it was. Whereas as it goes through this floral transition, everything gets a lot more organized and it becomes something that you actually can visually see is going to turn into a wheat spike. Okay, I don't know how I'm doing for time, but with that, I'd like to thank everybody that's in my group, especially Ian, Andy, and Vanda, who are working with me on the spatial side, certainly on the server side of what we do. The rest of my group are always really supportive, so I thank them. Sonia for all of the good conversations that we've had for troubleshooting this new technique together, and of course Katie Long for the samples and the collaboration we've had with the JIC. The email address for any inquiries for new projects is up there on this acknowledgement slide. And also I have a QR code if anybody's interested in learning more about the technique and how the MERFISH works. Okay, thanks.
Let's see. So hi everyone. My name is Sonia Fonseca. I work as a postdoc in editable topics group at the Earlham Institute in Norwich. And today I'm going to talk to you about my experience with using MERSCOPE with challenging cell types. And the challenging cells that I work with are megakaryocytes. And we are especially interested in the single cell and spatial transcriptomic analysis of the microenvironment. So to give you a bit of a background, megakaryocytes are a rare population of cells. They are mainly found in the bone marrow, and they only represent 0.1% of the cells, but they are responsible for producing more than 1 million platelets per second. And the interesting thing about this is that the regulation of such an important process is still not very well explained. And another very interesting thing as well is that although megakaryocytes are mainly found in the bone marrow, they can also be found in other tissues like lungs or liver or spleen under different conditions. But at the moment, the account, the origin, and the contribution to platelet production is still not clear. So megakaryocytes are a very special type of cells to work with. And one of the reasons is because they are really big cells. So they are bigger than other blood cells like B cells or red blood cells, and they are way bigger than the platelets they produce. And why are they so big? Because instead of following a classical mitosis for duplicating, as most cells do, they follow an endomitosis process. So it starts the same way, but then after duplicating the DNA, they do not split. They just get bigger and bigger, and they become polyploid with high numbers of DNA copies, up to 128 copies. So what do they do? When the megakaryocytes are mature, they form these structures that you see there called proplatelets, which are basically elongated fragments of the cytoplasm that enter the blood vessel and release platelets into the bloodstream. The main function of platelets, as you might know, is to form clots to stop bleeding at injury sites. But sometimes this platelet function can be dysregulated, and they can also form unwanted clots inside the vessels, which can be life-threatening. So producing platelets is the main function of megakaryocytes, but recent studies have also shown that in some cases megakaryocytes can function as immune cells. And this might be correlated to ploidy. So lower ploidy, they have more immune function, and higher ploidy, they have more platelet production function. But again, this is not very well understood yet. So as you can see, with such a heterogeneous population, bulk studies are not very informative. We need a single cell approach to be able to correlate size and ploidy with gene expression and function for each individual cell. So the first thing you need for single cell studies is, of course, to isolate single cells. For this study, we first started single megakaryocyte isolation by manual picking. So literally manual picking with a capillary under the microscope. So before we collected the bone marrow from mouse femurs, we enrich in megakaryocytes by filtration and then stain them with specific markers. And then manually pick with a capillary. The main advantages of this technique is that it provides valuable morphology information. It is a gentle technique, and it doesn't have a cell size limitation, which is really important for our type of cell. However, as you can imagine, it's quite low throughput and quite a slow method. There are several methods available for high throughput single cell isolation. But of course, due to the nature of the megakaryocytes, so they are big, they are quite fragile, irregular, and they often have attached other cells. Not all of these methods are ideal. For example, droplet-based methods like 10x or also traditional sorters can have limitations on cell size. And of course, they do not provide information about the cell morphology. We have also tested one of the new generation flow cytometers with image-based sorting, but again, they have problems with big and fragile cells like high ploidy megakaryocytes. And so here it is where MERSCOPE comes as a very useful tool for single cell analysis of this type of challenging cells. So not only to study the spatial organization of the microenvironment, but also we can analyze single cell suspensions, providing morphology information, avoiding cell size limitations, and in these cases, keeping the tissue sectioning and just doing a tissue dissociation or cell enrichment instead. So to obtain this bone marrow cell suspension, we just collected mouse femurs, extracted the bone marrow just by brief centrifugation, and then we loaded into a perfusion chamber attached to a specific MERSCOPE slide. And then we follow the Vizgen protocol for suspension cells to run it in the instrument using our customized 300 gene panel. And we are getting images like that, which are pretty images. So here, hopefully, you can clearly see how different they are, the high-ploidy megakaryocyte from other cells in size and shape and so on. And if we zoom in, we can see that we have information about single megakaryocytes, the specific transcript, single cell level. Of course, this is an ongoing project, but so far we have been able to identify specific markers for megakaryocytes that have been reported in bulk studies, but have never been reported before at single cell level. So I think it looks really promising. But of course, where we are really interested in is in understanding how the microenvironment influences the megakaryocyte function and how a disruption of this organization could lead to a failure in megakaryocyte maturation and also in platelet production. So here's an example just to illustrate the importance of the megakaryocyte microenvironment. So this is a bone marrow section, and hopefully you can see marked with yellow arrows where the megakaryocytes are. So I mentioned that they're a rare population, so they're not very frequent in the bone marrow, but because they are big cells, you can probably see that they occupy a large proportion of space. So the interaction with the surroundings for these type of cells might be even more relevant than for other cells in terms of influencing the different functions. So yeah, for example, just to illustrate some of the potential changes, for example, the proximity to blood vessels of the megakaryocytes might mean that they have access to more oxygen, more nutrients, so they can get bigger and produce more platelets. But also, the colocalization with different cells can activate one pathway or another. For example, exposure to pathogens in the lung, and probably just that, and not ploidy, could favor the immune function of the megakaryocytes. So we would like to investigate that. So to do that, we tested the MERSCOPE platform for mouse bone marrow sections, and we obtained images like this. So you can see in blue is the nuclei, and in green is the RNA staining, marking the cytoplasm. So the protocol worked overall, as you can see, but there are some areas that hopefully you can see at the bottom of the image that are quite out of focus. Probably this is due to sectioning conditions, as was explaining very well with the lifting of the tissue. We have many areas that you can see that they have single cell resolution, which is encouraging, but Unfortunately, the number of transcripts detected is quite low at the moment, probably due to low RNA quality in the sample, but also we think it might be because of the autofluorescence background we have with the bone. So at the moment, we are working hard trying to find a protocol to improve this bone marrow sectioning without compromising the RNA quality, of course, but also to remove or minimize the autofluorescence of this type of sample. So we hope that the new Murphy's chemistry that we haven't tested yet, but we hope that it can help with many of these issues. Because with the traditional Murphy's, the one we are using at the moment, for samples with low RNA quality or a high background noise, the signals could be masked. And so the transcript quantification is more challenging. But with the new Murphy's workflow, it minimizes these with an optimized probe structure. We will have better RNA hybridization, and also it will provide a stronger signal. So we are quite hopeful with this. In the meantime, we've been busy working with another very interesting megakaryocyte microenvironment, which is the lung. So as was explaining as well very well the workflow, we dissect the tissue, prepare the OCT blocks to cut in the cryostat to get 10 micron sections onto the slides, and then we follow the MERSCOPE protocol, in this case for fresh frozen tissue, and then again we run it in the instrument and we use the customized 300 gene panel. And we obtain images like this one, which is basically the whole section with single cell resolution. We have the DNA again in blue, cytoplasm in pink, and cell boundaries in green. Yeah, again, after performing the cell segmentation, we can assign transcripts to individual cells, and we can generate UMAP to clusters and to cluster and annotate different cell types in the tissue. So if we select one of our cells of interest, in this case a mature megakaryocyte, and we zoom in, we can see now this the megakaryocyte microenvironment with single cell resolution. And because now we have the information about the annotation of the cells, we can now overlay the annotation of the neighboring cells, like this one. And we can now begin to study cell-to-cell interactions in a more specific way. So at this moment, we're just starting to analyze these lung data sets, but we already see the potential to find mechanisms that regulate megakaryocyte maturation. So, for example, we have seen that although TLR2 is mainly expressed in alveolar macrophages, probably due to the exposure to bacteria in the airways, some specific megakaryocytes, but not all of them, are also expressed in TLR2, which has been reported as significant for megakaryocyte maturation. So we think that analyzing the microenvironment might be crucial to understand these differences in gene expression and eventually in megakaryocyte maturation and function. And another example, we have also noticed that the presence of some cells with potential influence in megakaryocyte maturation, like M2 macrophages, we have noticed that they are always in the proximities or in contact with mature megakaryocytes, and this interaction has not been very well defined yet. So now we can start exploring these or other potential influences in more detail. So yeah, in summary, we've seen that combining single cell expression with the spatial information is helping us understand megakaryocyte development and function with more resolution, especially with more context than ever. We can now open the door to new research. For example, if we understand the regulation of the physiological polyploidy of megakaryocytes, we can maybe help establish a baseline model for pathological polyploid states like cancer. Of course, this can help also to treat or prevent platelet-related conditions, and eventually probably refine the current in vitro platelet production and coming closer to a donor-free platelet production. And yeah, thank you very much to everyone involved in this project. Everyone is really, really helpful, especially my PI editor, Ashley, a lot of work, Ian, everyone in the group involved with this. Yeah, thank you very much for the help. And I think that's all for me. Thank you very much.
Thanks, Sonia, for the beautiful talk. Thanks, everyone. We still have 10 minutes to address some questions. We've had quite a few questions in the chat, which is really nice. So yeah, thank you, all three of you. These talks were amazing. And for me, it's really nice seeing this like beautifully structured summary of all the work you've been doing for the past, year and a couple of years. So we've got a few technical questions. I can maybe read out the questions and then Ashley and I, or like Sonia and I, or the three of us can like discuss it. So about photo bleaching, Ashley was mentioning that she photo bleached during clearing, and someone was asking why not before, during permeabilization. Did you try different things?
Yeah, so the good thing with Vizgen platform is that we can do it before or after, which is really, really nice. So what I tend to do, I quite like that I get it out of the way. It fits better with the workflow, that I do it ahead of the week that I need before the run to do sort of more like washes and liquid processing. So it's nice that I do what is actually 8 hours of photo bleaching, which is quite extensive for a lot of tissue.
Bit of echo here. Hello?
Hello.
Okay, no, I think it's sorted. Yeah.
Yeah, so for plant tissues, it's quite extensive, the amount that we have to do. So actually, that's really a whole day. So to get that out of the way ahead of everything else is really nice. But what I then do before a run, before I stain, is I just check that that level is as good as it can be. And I give it an extra top up sometimes of 3 hours before a run, before I'm doing the staining. So it's nice that I've got the flexibility to do before or after. Yeah.
I think it's really good that you implemented checking on the microscope as well. And that's something that I've been recommending to some other users as well, because it's just a really quick, efficient check.
Yeah.
We haven't really compared if there's any like difference before or after clearing. I suppose that if you do it after clearing, there's less like other types of fibers that could diffract the UV from the photo bleacher. But I would think that this is quite a minimal impact because we get, equally good results overall. With both processes.
I think that what they're alluding to is maybe that there might be digestion that might aid that autofluorescence, and we did try that, but really it didn't make, for this tissue type, it didn't make any real difference to do it after clearing. So yeah.
Okay, awesome. The next question was, when you were showing Katie's data, someone observed that the transcript kind of co-localized around the cell boundary, and they were asking whether it could be an artifact or if it represented a true biology. And you were saying that you didn't observe this in other tissues. I think this is something that could be really easy to check by typically either looking at the raw data and seeing if you get any sort of like parasite signal on the edges. I remember that for this data set, the autofluorescence from the cell wall was really minimal and did not interfere. And also in terms of cell structure, the volume occupied by the nucleus was quite huge. And there was like literally just like a big bore nucleus and then like a little bit of cytosol and then the edges. So I would err on the side of saying that it's biological, but I think that's something that could be really easy to check by looking at what's expressed and if there's any like polarity. But yeah, I think it's mostly something about space and these transcripts being more cytosolic than nuclear, I would say.
Yeah, so wheat is a polyploid. It's a hexaploid actually, so that nucleus is going to be quite massive.
Yeah, it's huge. We had someone asking, can you do 2 different plant species like wheat and Arabidopsis on one slide? Yes, you can. As long as the homology between both organisms for the genes in your panel is not too important. So I usually, I'm kind of like the annoying person being like, you have to check every sequence because you actually need to align them and check that there is like a sufficiently long, unique sequence. Like if you have really nucleotides interspersed everywhere, it's not going to work properly and you will have, poorly decoded or hybridized transcript in both species. So you just need to make sure they're different enough. And you can get verification kits for the 2 different species separately. You won't be able to get a verification kit for both, just due to, you know, this is a really cheap kit. We don't do any imaging cycle. It's kind of like a quick, you know, control. So we are not able to accommodate having several species within a verification kit.
Yeah, that's right. Yeah. So I don't know if it's nice for you to explain, is that your bioinformatics team, as I understand, they will go through a lot of that alignment and checks with you, right? So you can do your own pre-checks, but Vizgen are really good at advising you about that, right?
Yeah. We have a dedicated, for all custom panels, we work with a team of bioinformaticians, and they're going to review your targets, you know, the gene abundance, the sequences. So there's a lot of support provided there. Just looking at all the questions. We have a question about plant tissue, with someone noticing a lot of tissue lifting. If it happens during the washing steps after the photo bleaching, but before gel embedding, any tips to avoid this? We tend to recommend drying the tissue as much as possible, even if it's not, you know, like at the steps you expect to. I think, Ashley, you struggle with a similar problem. Would you mind talking about your experience?
Yeah, so it's good that you're making those checks. Sometimes we're now more used to the effects, like the subtle effects that can cause that at that stage. So, you know, after you've really committed to going through the workflow. So what we do is we check after sectioning, just sort of scan across the whole slide and look for any different effects of sort of a Z-sac difference of your tissue, which just shows like a very, very subtle lifting. You know, it's in the microns, but that's enough to mean that you won't have resolution of your probe spots. So I don't know if that helps, you know, before you get to that stage. But yeah, I think Carol's right, is just dry it out really a lot more than ordinarily you would think is correct for the way that you would ordinarily treat RNA. It feels very, very alien to us to be treating it like this, for sure. Those of us that do sequencing ordinarily. So yeah, trust it, it works.
There's also some people also use poly-L or poly-D lysine coating on the slides. So that's something that you can do when you get the MERSCOPE slides, treat them with that. And with some tissue types, it helps with the adherence. It really, because this is like more of a chemical, I think it helps more if it's like tissue that's lifting due to high fat content. So I think plants, they lift just due to like the structure and the rigidity of it. So it would need to be tested out. We have 2 related questions. Can the photo bleaching negatively affect your RNA quality? And someone who said they understood photo bleaching was done before fixing. It's done after fixing. That even if you use fresh tissue, you'll have to do a post-sectioning fixation before you do anything. So as the moment you take it out of the cryostat, it needs to get fixed. Otherwise, yeah, of course, your RNA will degrade. So once the tissue is properly fixed, and as long as it doesn't dry out too much in the photo bleaching at that step, so it doesn't get caked in clearing solution, it won't affect your RNA quality. We have a question for Sonia. Someone was asking how many transcripts per field of view did you get, approx, and how many transcripts per CD8 positive T cell were you able to get in the mouse lung experiment, if you remember?
Yeah, I was going to say, I don't think I remember the specific for that particular cell type. We did detect them, and it was an important population, definitely. And it was like throughout the whole lung, as expected. That's what I remember with the imaging, but I don't think I have the number in my head. About the transcript per field of view, you have to bear in mind that it was not specifically designed, this panel, for lung at the moment. At the minute we were designing that, it was more focusing on bone marrow. So overall, the transcript per field of view, it was not as high as the instrument can provide. So it was not like to the top of what we can get out of it. It was good. It was uniform, and we were happy with the results. It was enough to study our to start studying our actually the megakaryocyte microenvironment. But yeah, in the future, if we got through this pathway, maybe we can start doing more specific for different microenvironments, like as Ash was suggesting, for example, in a 300 gene panel, 100 genes can be more focusing on lungs, 100 can be more focusing on bone marrow. So yeah, that's something to come. But yeah, I don't think I have the numbers in my head at the moment. But yeah, contact me and we can have a chat maybe. Thank you.
Yeah, that's a really good point. I also want to add, if you are struggling to get high counts from tissues that have a lumen, like lung, mouse colon, something that, I don't know, probably you're already doing it, but something that really helps is upon dissection, you can immerse the tissue in VRC, so vanadium ribo whatever. I don't know, it's an RNase inhibitor, but like a chemical one. And so if you immerse it in that, so dilute it in your medium for like half an hour, it really helps stall RNA degradation. Maybe some people also are perfusing the animal with PFA, but while it's good, sometimes it can be a bit slow. So for this kind of organs, so everything that's like exposed, lung, skin, colon, I would really recommend doing this treatment. I can give you more information, of course, if you reach out. And do we have time for one last question, perhaps? Or because someone was asking, thank you so much for these interesting talks. They have some doubts regarding data analysis. Have you tried to combine MERFISH results with mass spectrometry imaging analysis, so metabolomics or proteomics? I don't know if that's something that you guys do in the Earlham.
No, sorry, we can't comment on that.
No worries. Great. I think I've gone through most of the questions. Thanks everyone for attending. That was really informative and I really enjoyed this. So thanks again, Ashley, Sonia, and Ian. That's great. And yeah, please feel free to reach out to us or to the Earlham team for any support. Lovely. All right, take care, everyone.
Bye-bye.
Bye.